Well, hello, everybody, and welcome to the Faraday Institute. Good morning and good afternoon or good evening, depending on where you're watching from today. Especially warm welcome to you if you've come here in person. We have a real live audience here in Cambridge, and you're all very welcome to today's seminar. This is the third in our regular series of Tuesday seminars. And as always, we welcome your questions and comments after the talk. So I'm Paul Ewart. I'm the new director here at the Faraday Institute, and it's a pleasure to be hosting this event. So I will channel your questions to our speaker online, and if you're here in person and you want to ask a question, I'm going to ask you to come to the front to the podium on my left and pick up the microphone and put your question then. But today we're delighted to welcome Professor Jeff Hardin, our speaker today, and to thank him for getting up so early as he's speaking from Wisconsin in the United States. So Professor Hardin is a chair of the Department of Zoology at the University of Wisconsin-Madison, and he holds degrees in theology and in biophysics. He's very active in promoting the dialogue between science and religion, and in particular the work of the InterVarsity Graduate Christian Fellowship on the University of Wisconsin campus. And as well as being an internationally recognized educator, Professor Hardin is also a highly respected research scientist in the field of genetics, cell and embryo development, work that has extremely important implications for understanding birth defects in babies and also in cancer. So he's extremely well qualified to speak to us today on his subject of self-made people, the emerging embryo, biology and belief. And I'll hand over to Professor Hardin now.
Great, thank you, Paul. Well, it's really a privilege to be with you all this morning in the central part of the United States. And I am always delighted to do anything that I can with the Faraday Institute. Paul, congratulations on your new role with Faraday. It's such important work, and I just hope things go very well. I've been involved with a number of projects with Faraday. Ruth with Faraday Churches has been absolutely amazing, and she has stoked my sense of wonder about the embryo, something I'll be talking about today. I've had a little bit of involvement with Faraday Kids, such exciting work going on there, and I'm involved with an organization called the Biologos Foundation here in the United States, which in some ways is a sister organization, some ways not, to Faraday, and so it's always a great privilege to join with you. Today, what I want to talk about is embryonic development, and that's what I do for a living. And I want to talk about some of the things that we're learning about embryonic development, and then at the end, I want to talk about some of the potential implications. So where are we headed? First, I want to talk about, and I've stolen a phrase from Ruth here, embryos as wonders of the living world. And I want to talk about embryos as self-made systems. Next, I'll talk about a peculiar but really fascinating property of embryos, their resilience. And then I want to talk about embryos as personal stories, because each one of us started as an embryo. So first, let's begin with a bit of wonder. I have two kids, 34 and 32. This is my 32-year-old, well, let's just say a long time ago, my son, Christopher. And if you're a parent or you've had the privilege of holding a baby in your arms, you can't help but be captivated by how amazing the processes of embryonic development are. How remarkable it is that these processes lead to such incredible reproducibility, wonderful digits, fingers, toes, facial features, all of the things we associate with a newborn like my son Christopher here. Building an embryo, building an organism from an embryo is a staggering challenge. There's a lot of things that go on. We start as a one-celled zygote, as you see on the upper left, and then some complicated things imaginatively shown by the white arrow here occur. And then we get this incredibly complicated organism out at the end. And a human infant like Christopher here in this picture has hundreds of cell types and about 5 trillion cells. So the task at hand is a daunting one. But developmental biologists like me are trying to understand biologically how this process occurs. So let's just remind ourselves of some landmarks in embryonic development. First, of course, is fertilization, and that's a very contingent process. If you were started your life as a zygote in the typical way, a single sperm has a 1 in 250 million chance of fertilizing an egg, but only one was successful, leading to you. So it's a contingent process, but fertilization kicks in physiological and other changes in the zygote, the fertilized egg. And ultimately, this leads that fertilized egg to divide. So here's a mouse embryo. Looks very similar to a human embryo, undergoing the early divisions known as cleavage divisions in the embryo. And you can see that initially the cells look like a loose cluster of grapes, but then they become very compacted, and then a hollow space forms in the middle filled with fluid. That's called the blastocele. There are a lot of complicated things at the molecular level going on during all of this, but ultimately you get this structure known as a blastocyst. The cells at the top are called the inner cell mass, and they're very special. They're what becomes the baby. So that group of cells is what formed you and me during embryonic development. And those cells are uniquely and extremely developmentally flexible. We'll come back to that. Well, eventually multiple layers form in the embryo, and some of those layers have been surgically removed here in this human embryo, but you can see a line running from top to bottom, a kind of a dark smudge. That's called the primitive streak, and that forms via movements during the process known as gastrulation, which is cellular choreography on an absolutely massive scale. We probably understand vertebrate gastrulation best in this organism. This is the African clawed frog, Xenopus laevis. As I run this forward, you'll see that there are all kinds of crazy, complicated things happening. Cells are moving, cells are rolling around this circular edge called the blastopore into the interior. The embryo is elongating, and by the end of this movie, the anterior end at the top is different from the posterior end. So the top and the bottom, the head and the tail of the embryo are well defined by the end of this process as the embryonic axis, body plan of the embryo emerges. This happens in mammals as well. Here are some mouse embryos. They've been genetically engineered so that some of their cells are glowing. And you can see on the left that there's a group of green cells, which is elongating top to bottom. That's along the head to tail axis. There's a different genetic engineering feat accomplished in the embryos in the middle and on the right. In their case, it's the pink cells that are doing the same thing. And you can see that there are rows of tissue that are getting very long and moving very dramatically. And that's a hallmark of gastrulation. This is incredibly complicated choreography, but it's choreography that is written on the fly as these embryos and the cells that constitute them move about in very dramatic fashion. Well, eventually the central nervous system forms, and this is what we all looked like. We essentially looked like a burrito. That's what I think of in the US anyway, when I look at this. And that's because a tube has formed called the neural tube, which is going to form the spinal cord of the embryo during the process known as neurulation. We probably understand this the best in amphibians as well. These are some axolotls, so salamanders, and you can see that there's a very dramatic set of folds that form, and they look like a keyhole shape. And then eventually these folds meet at the middle on the top of the embryo, which will form the back or dorsal side of the embryo. And then the anterior part at the top eventually closes over. That's what's going to form the brains later as this tube becomes more and more complicated. And that's part of something known as organogenesis, making organs. And during this stage of development, a lot of the standard organ systems that you and I are familiar with take shape, as well as formation of things like the limbs. And you can see that those are forming in this beautiful picture of a human embryo. Well, that's the wonderful aspect of embryonic development, but let's talk a little bit about how this works. One of the great privileges of someone in my kind of, well, I'll say seniority stratum is that we have made dramatic strides in the course of my career in understanding at the cellular and molecular level how embryos make themselves. So let's talk a bit about that. You don't need to be a modern developmental biologist to have thought hard about this. In fact, Aristotle, really the first great natural historian in the West, cracked open bird eggs and looked at them as the embryos inside of them were developing. And you can see that there's a chicken embryo developing, floating on a large yolk mass here. And Aristotle realized that there was something going on that involved the interactions of simpler parts that led to progressive complexity in the embryo. And he called that epigenesis. Now that's different from the chemical modifications of DNA that are part of the modern field of epigenetics. But it's this idea that complexity arises through the interaction of simpler parts. Now for Aristotle, he felt he needed to invoke something called entelechy, an immaterial organizing principle that needed to run the show, provide the metaphysical glue for this process. Modern developmental biologists tend not to find that to be a helpful hypothesis. I think if Aristotle were alive today, he might be a developmental biologist. He was a remarkable observer of nature. Well, what are some big ideas in developmental biology that we need to come to terms with? Well, one is something called differentiation. In other words, how do parts become different? Another big idea is morphogenesis, the building of the body, from the Greek word morphe, which means form. So these two processes are the core of developmental biology, and understanding them in the various contexts in which they occur in an embryo is what biologists like me are about. Now, it turns out that these processes go hand in glove. Different. They can send signals to nearby cells, or they can move, and this allows for building certain parts of the embryo at certain times, and those new positions then allows them to talk to other cells that they wouldn't be able to talk to earlier, and so on and so on. So development is a progressive spiral of differentiation and morphogenesis going hand in hand. Differentiation involves changes in the expression of genes, bits of DNA that provide the information to make RNA and protein molecules. And one major achievement of modern developmental biology is the discovery that there are banks of protein switches or gene regulatory networks that regulate a whole host of genes that are associated with a particular differentiation event. So if you like, you can think of gene regulatory networks as a bank of switches. In some cases, they activate the expression of key genes whose products are crucial for the next step in development. In other cases, they shut down the expression of genes that are no longer needed to be expressed. We know a lot about how all of this works. These proteins hop onto bits of DNA, regulate whether those pieces of DNA are accessible to the machinery that allows their information to be read out and proteins to be made from those particular genes. So these gene regulatory networks are a feature of multicellular animals or metazoans. They're well conserved. So once they were invented over the course of evolutionary time, some of these gene regulatory networks are stubbornly persistent, very stable against perturbation, but they're very useful because they lead to incredible reliability of developmental processes. So in that way, gene regulatory networks are sort of plug and play circuits that can initiate a developmental module. This leads to differences in cells. The cells acquire certain characteristics, and then they do those processes that I've talked about already, known as morphogenesis. The most famous example, perhaps, is the homeotic complex. These are a set of protein switches that help to establish the identity along the body axis from head to tail of animals. They were first discovered in fruit flies, but they are found in all metazoans, including mammals like the mouse on the bottom. And they are conserved. They are important for specifying body parts. And in the mouse, or in the fly in particular, you can get really dramatic transformations of body parts when these don't work properly. So this is a great example of a gene regulatory network. Now, I don't actually study these. And I want to argue that the other part of development, morphogenesis, non-genetic regulatory processes, are every bit as important as these. Here's my colleague, Sean Carroll. He wrote a wonderful book called Endless Forms Most Beautiful. Now, he actually made a living studying gene regulatory networks, but even Sean recognizes this. And he tells the story of embryologist Paul Weiss, and he says, Paul Weiss once illustrated the reductionist dilemma for his fellow embryologists. He showed a picture of an intact chicken embryo, then an embryo that had been put through a blender, and finally an embryo whose blended components had been centrifuged together. Blech. Weiss bluntly stated the problem for reductionists: how to get that chicken back. Sean goes on to say, our toolkit genes, gene regulatory network genes that encode these switch proteins, are just pieces of our genetic material. A small fraction of the 14,000 or so genes in a fruit fly, or the 15,000 genes of a mammal. Granted, we've identified many critical pieces, but the question remains, how do we get that chicken back? And that involves cells doing things in sociological context together to build the embryo. Now, I don't know about you, but my older son, John, loved Legos, and maybe you guys have uniquely British instantiations of Lego building kits, but here's one. And you can get a great structure as you see on the lower right. And it's built from the pieces that you see on the upper left. But I think we'd agree that although you can see that there's differentiation occurring in the pile of pieces on the upper left, there's a door, there are bricks of different colors, different dimensions, etc. I think you'll agree that there's something fundamentally missing, and that's that they're not organized properly. They have to be built. So indeed, some assembly, actually a lot of assembly, is required. And that's what I want to talk about next. Clearly, that self-assembly involves mechanics. So my PhD is in biophysics, and I really thought hard about these kinds of mechanical issues, but I was not unique in this regard. Here's Wilhelm His, 19th century anatomist. He said, to think that heredity will build organic beings without mechanical means is a piece of unscientific mysticism. Wow. He wrote a really influential book called Unsere Köpfe und das physiologische Problem ihrer Entstehung in 1874. But we've become more sophisticated in thinking about these topics. Here's one of my favorite examples. This is work from G.G. Selman from 1958. He used tiny little barbell-shaped magnets and put them on the neural plate, that part of the central nervous system, before the folds elevate and close over the structure to form a tube. And he used an electromagnetic field to push apart the neural folds to measure the force of closure. Wow, remarkable. It's hard to think about that using genetic wiring diagrams. You have to come to terms with physics. Nowadays, as you see this example from Oker Kampas's lab, we use fancy things called ferromagnetic fluids and fancy microscopes to do more sophisticated versions of Selman's kinds of experiments. This is just one of a number of physical and cellular mechanisms that are common in animal embryos. And this is what really got me hooked as a graduate student at the University of California, Berkeley. In my case, in particular, I studied a sea urchin embryo, and I studied gastrulation in sea urchins. And I studied how this tube on the right elongates. So this is called the archentron. It forms the primitive digestive system. We had to do the same thing as embryos. And I did mechanical modeling to try to understand this a very long time ago. All right, so what are the kinds of modules that we could think about that shape embryos? Well, I don't want to take you through all of them, but there are many different kinds. Modules that allow cells to clump, to stick together. Modules that allow cells to know where they are in space. Modules that allow cells to make crawling doodads, protrusive appendages that allow them to migrate. Modules that turn off cell adhesions selectively, differentially. Modules in which cells exchange diffusible proteins that act as signaling molecules. And these are what Stuart Newman has called dynamic patterning modules. they execute programs or events that don't involve these genetic switches. They happen after these switches have done their activity. But we can recognize a limited repertoire of these dynamic patterning modules in embryos. Now, Stuart Newman wants to propose that there's something additional going on. That's what he calls inherency. And this is the idea that if the conditions are right, these modules will just snap into action, and that they are kind of a latent tendency waiting to be realized when conditions are right. And that might be a little more controversial idea, but it's certainly true that these modules show up over and over and over again in different contexts in different animals, but they're very similar in each animal that we might look at. One of these that I've studied is cell adhesion. So I understand this is the common adhesive on the left in the UK. We don't have this one over here in the US. I guess we're impoverished, but there are molecules that carry out stickiness programs in embryos. And one of those is one we've worked on called the cadherin complex. It's a molecular glue that allows cadherins sticking out of one cell to interact with cadherins sticking out of another cell to allow them to attach. And cell adhesion as a developmental patterning module is very ancient. The first multicellular animals have cadherins, and I'll show you that in fact things that are even simpler still have them. Now I happen to study the nematode worm C. elegans, so my favorite cadherins are in the red box here. Another module is one that's involved in neurulation and sometimes in gastrulation. And that is that sometimes cells have to pinch off their ends, and this causes an in-pocketing of a tissue. That's something called apical constriction. And this happens in the amphibian neural plate during the formation of the central nervous system, as you see on the left. But it also happens in my favorite organism, the nematode worm, as it is internalizing cells, the green cells in this case on the top, something that we've worked on. Another one is something called convergent extension. Cells engage in something sort of like a line dance. Think of those period piece Jane Austen BBC adaptations where people are in a line and then they change positions. That's maybe how to think about convergent extension. So on the left, the orange and the blue cells wiggle between one another to make a single row, but much longer. So here's an example from my lab where I've colorized 4 cells, blue and green, and you can see them moving and they wiggle between one another. And this happens over and over and over again in lots of different animal embryos. One of the things that's required is a way for cells to know where they are in space. And one such module is called the planar cell polarity pathway. And there are lots of proteins involved in this. But when these proteins are present and you have a group of cells, often this pathway spontaneously or at least preferentially kicks into play. And if this pathway is not operating, here's another embryo from my lab, you can see that bad things happen. So now the yellow and the green cells are not in a strict alternating pattern. They're trying to move together with their mates. That's something that shouldn't happen. So all of these patterns, these developmental patterning modules help to drive what happens after these gene regulatory networks initiate differentiation of a group of cells. And it's probably the fact that these physical chemical mechanisms are so robust that accounts for another, I think, amazing property of embryos. That's their resilience. The most famous experiment, in my opinion, along these lines was done by this fellow, Hans Triesch. Hans was working on sea urchin embryos. That's how I got my start. I happen to have a German degree. It's one of the few areas where my German degree is useful. It's for reading the embryological literature from the 19th century. Triesch, a really clear writer. He did a mind-blowing experiment on sea urchin embryos. You can remove calcium from seawater and cause the cells of an early sea urchin embryo to fall apart. And Triesch separated the 4 cells of a 4-cell sea urchin embryo. Now, instead of getting 1/4 of an embryo, he got a completely different mind-blowing result. And that was that each of these 4 cells could make a perfectly proportioned little sea urchin larva called a pluteus because of the easel shape it reminded embryologists of. This blew Driesch away. He couldn't understand it. He called this a harmonious equipotential system. Embryologists like to dazzle you with terminology at times. What he meant by that was that in the face of such drastic perturbation, the cells figure it out. They find a way to develop. In other words, injury reveals cryptic resilience in these embryos, and it's probably these developmental patterning modules now being engaged in new ways that they wouldn't in an intact embryo. Now, Driesch thought there would be no way we could really understand this. He stopped working on embryos, and instead he became a vitalist philosopher. So he proposed that, in fact, he used the word that Aristotle had coined, entelechy, to talk about an organizing principle that drives the embryo forward in the face of adversity like this. I think most 20th and 21st century developmental biologists have not found these concepts to be especially useful in thinking about biological mechanisms. But I think Driesch was onto something, and that is that however we seek to account for this, we've got to try to understand what have we done to the embryo, what's been initiated, and how is it that the embryo has this latent capacity for self-healing in this way? That's what I mean by resilience. Now, in the case of sea urchin embryos, I think you can make an argument that the ability to respond to injury reflects some modules that, in fact, are cryptic. So you may not know this, but sea urchin larvae, in the presence of predators like fish that might eat them, can undergo a non-embryological form of reproduction. They can undergo asexual cloning, and they respond to mucus in the seawater that the fish secrete from their skin. So the larvae on the left, the little round structures, are a forming baby clone, forming in a completely different way from normal development. So having this cryptic ability to do things in a different way has some selective advantage for these sea urchin larvae. And they form these little mini plutei, as you can see on the right. They're much smaller than normal. So there's more ability than we know within organisms to survive these kinds of insults. And these dynamic patterning modules that I've been talking about, this resilience in the face of changes in conditions, is very ancient. Let me tell you about one example. This involves organisms that are the sister group to multicellular animals called choanoflagellates. They're called choanoflagellates because they look very much like a cell type that is found in the poriferans, marine and freshwater sponges. They have a little collar and they have a long flagellum, which allows them as single cells to swim. But some choanoflagellates can, when they want to or when they need to, organize into multicellular groups. So they have facultative multicellularity. They can make multicellular structures. This is work that's been pursued especially by Nicole King's group at University of California, Berkeley, so that these cells can form on command under certain conditions a rosette. So that looks an awful lot like a dynamic patterning module. And in fact, it involves the pink stuff. This is extracellular protein that acts to hold these cells together into these rosettes. I think more surprising to people like me was that although these choanoflagellates are not multicellular, they make a large number of cadherin proteins, that cellular glue I talked about earlier. And it's found in particular places within single cells. But choanoflagellates, at least some of them, have more than 20 of these proteins. So they've got the ability to aggregate into multicellular groups, but they don't always need to invoke it. More recently, Thibaut Prunet, who was the postdoc in Nicole's group, found that some choanoflagellates respond to light, and they can form sheets of cells. And then when you put them in the dark, they can turn themselves inside out. They do this so that when the light is on, they can feed, but when it's off, they can swim. And so the group on the right, I think, is here it goes. Watch this. It's going to turn itself inside out. Right about now. Here it's starting to curl. Whoa. Think about your umbrella on a windy day and you've got the idea. Now, what's really interesting here is that the same machinery that causes cells to pinch off that I talked about earlier and cause bending of sheets is in play in this group of cells that allows them to turn themselves inside out, but only under certain conditions when the light is off. So these are all examples that dynamic patterning modules were present in the predecessors to multicellular organisms in evolutionary history. And they remained with us as embryos, and they reveal themselves when the embryo is damaged. So that's true for us as well. So I don't know if anyone in the audience is a monozygotic, a so-called identical twin, but if you are, you'll know that you formed from a single embryo that split at some point during early development. This is a beautiful picture of these amazing girls. That's one example of the resilience of human or other mammalian embryos. Here is the one I find most mind-blowing. So you can isolate mouse embryos away from their protein coats, and you can stick them together. When they start producing cadherins at the 8-cell stage, the cadherins will cause the 3 embryos to congeal into a single embryo, which forms a single blastocyst, as you see on the right. So here are the 3 embryos, and then they combine to form the embryo on the right. And this is something called a chimeric mouse. And here is a chimeric mouse on the bottom. It has 3 different coat colors. Why? Because it was created by doing the experiment on the preceding slide. This is another example of the tremendous resilience of embryos, including ourselves. And in fact, this works in primates. So in 2012, the same experiment was done with macaques. And you can see here's one of the resulting embryos. This monkey resulted from the combination of 6 monkey embryos in this very similar kind of chimeric experiment. So we are capable of tremendous resilience as well as mammals and as primates. Now, a little later, I told you we would come back to this. This is what you and I all looked like, each of us looked like. This is a blastocyst. The cells at the top are the inner cell mast cells. And I told you that that's what makes a baby. So you came from inner cell mast cells when you were a blastocyst. And these cells retain tremendous developmental flexibility. They can't make everything, but they can make all of the cell types that form the baby proper, the embryo proper, what becomes a baby. And that means there's something, they have the property of what's called pluripotency. It turns out you can force cells by invoking a gene regulatory network to become pluripotent in this way. And if you figured out how to do this, and your name is Shinya Yamanaka, you got a piece of a Nobel Prize with Sir John Gurdon, who has been based at Cambridge for most of his career. And Yamanaka realized that 4 proteins were all that was necessary to turn a garden variety cell, say something that would normally form skin, into one of these very special cells. And these are now all called Yamanaka factors colloquially by people like me who teach developmental biology. These are all transcription factor proteins, and forcing cells to make them causes these cells to generate a genetic switch, a gene regulatory network that causes pluripotency in turn. And these cells behave very much like inner cell mast cells. that they can be directed to make many specialized cells. And lots of implications here for regenerative biology, regenerative medicine, those kinds of things. But I want to talk about a particularly mind-blowing capacity that these cells have. That is that if you put them in the right circumstances, they can self-organize, leading to differentiation and morphogenesis in ways that are strikingly embryo-like. So here's one such example. These are called gastroloids. The embryo on the left looks surprisingly like a mouse embryo. Mouse embryos are shaped like a rugby ball or an American football, turned inside out relative to human embryos and wrapped around the pointy end of one of those sports balls. And that embryo, or that embryoid, that embryo-like structure on the left, is doing many of the things that a mouse embryo would do. On the right is an embryo whose body axis, or what looks like a body axis, is forming. So these are called gastroloids. They're not really a gastrula. They're not quite like a normal embryo. But the ability for self-organization in these pluripotent cell aggregates is remarkable. Now, one of the groups that was working on this said this: we have to be careful using the term synthetic human embryo. They were working with some human cells because some people are not happy about it. And that's probably right. There are many ethical issues that we might want to consider. We could talk about these in the Q&A if you want. But I want to focus on the amazing self-organizing capacity of these groups of cells. Now, this is something called an blastoid. And this was formed by geometrically constraining 3 types of cells and just letting them do their thing. And it looks incredibly similar to a blastocyst. Remarkable. So this is an example of what George Church and his colleagues at MIT have called sheaths, synthetic human entity with embryo-like features. So whether sheath may safely graze here, I'm not sure. There are lots of questions that we could ask about these kinds of constructions. But one thing is clear, they're going to be useful model systems for trying to understand some of the cellular processes that lie behind these developmental patterning systems that we've been talking about today. Remarkable. All right, so far we've talked about the processes of embryonic development are amazing. And we can understand them at a cellular and genetic level. And embryos, because of these dynamic patterning systems and gene regulatory networks, show remarkable resilience when perturbed. But now I want to talk a little bit about the wider implications of this for you and me, because after all of us went through the journey on this slide. Admittedly, not all of us became my son Christopher, but we all started as a one-celled zygote on the upper left, that underwent cleavage divisions, formed a blastocyst on the upper right, eventually formed that burrito-looking structure on the bottom left, a neurula stage embryo, underwent organogenesis, and we were born. So there's a continuity to development. That's the first thing that I want to emphasize here. We all started as that one-celled zygote, and we can trace our life history through all of the developmental changes that are shown on this slide. But I'd like to think a little bit about this from a Christian perspective. I happen to be a Christian, and so I think about it from within that context. And here's a great father of the Protestant Reformation, which was recently commemorated earlier this month, Martin Luther. As he thought about embryonic development, he said this: the reproduction of mankind, of humankind, is a great marvel and mystery. That God consulted me in the matter, I should have advised him to continue the generation of the species by fashioning them of clay. As I should have also counseled him to let the sun remain always suspended over the earth like a great lamp, maintaining perpetual light and heat. Now, Brother Martin had his tongue in cheek here. What he was saying was that he probably trusted God to do a better job of running the universe than he would have. But he brings up an important point, and that is that we need to take embryonic development on its own terms. And Christians ought to have an even healthier respect for biological and other scientific processes because of their Christian faith. Let me talk about a couple of issues, though, that might come to mind as you think about what we've talked about today. First, I'll call deconstruction. Now, that means a number of things in today's modern climate, I realize. But this is a concern of the practical theologian in my household. This is my wife, Susie. She would say, life's a miracle. You're dissecting it. What are you doing here? Haven't you taken all of the mystery out of the process? John Donne said it this way centuries ago. The new philosophy calls all in doubt. The element of fire is quite put out. The sun's lost in the earth, and no man's wit can well direct him where to look for it. 'Tis all in pieces, all coherence gone, all just supply and all relation. So Donne was concerned about what we would have at the end of our dissection process. And that's a valid concern if our only intent is to dissect. But if our intent is to use non-human model organisms to understand the amazing properties of all embryos, I don't think we need to stop with deconstruction. Charles Kingsley, a 19th century theologian, said it this way: are we to reverence him less or more if we hear that his might is greater, his wisdom deeper than we ever dreamed? We knew of old that God was so wise that he could make all things, but behold, he's so much wiser than even that he can make all things make themselves. Kingsley's statement here was talking about Charles Darwin and what we were learning about evolutionary processes, but the same is true for the self-organizing processes that underlie embryonic development. In my mind, I have cause to reverence God even more when I understand the amazing biological processes that underlie what we see before our eyes under the microscope as embryos develop. There's another problem that we might consider as well, and that's alienation. Embryos are small. This human embryo is about 0.5 millimeter in diameter, maybe a little bit less. Very tiny. This is not a new problem, of course. I'm of a certain seniority stratum, shall we say, that I remember the moon landings in the US Apollo space program. And this is a shot taken by Apollo 8 from orbit of the moon back at Earth, called Earthrise, a very famous photograph. And these kinds of images shape how we think about our planet. We see our planet in a much more fragile way now than we did before these kinds of pictures were available. And certainly this is very relevant given modern discussions of climate change and the climate crisis. The same is true for embryos. When we look at embryos, they're tiny. And we might think that because of their diminutive size, they have diminutive significance. They're insignificant. But actually, this is not a new problem. The ancient Hebrews were thinking about this millennia ago. Here's a poem from the Hebrew Old Testament. When I look at your heavens, the work of your fingers, he's talking about God, the moon and the stars that you've established, what are human beings that you're mindful of them, mortals that you care for them? Yet you've made them a little lower than God and crowned them with glory and honor. You've given them dominion over the works of your hands. You put all things under their feet. This poem, Psalm 8, is musing on the vastness of the cosmos and our insignificance. And yet it concludes that size is not determinative of significance. And that's maybe a good way for us to think about this particular problem. You see, God knows our whole story. The Bible talks about humans as being begotten gifts, precious. It values the weak. And it understands that there's a continuity between embryos and adults. And that God had us in mind all along, throughout the trajectory of our lives from zygote to baby to who we are today. And in fact, here's another Hebrew poem that I quote in my developmental biology class. It's from Psalm 139. It says this: you formed my inward parts, you wove me in my mother's womb. I'll give thanks to you, for I'm fearfully and wonderfully made. I'm amazing in my construction. Wonderful are your works, and my soul knows it very well. My frame was not hidden from you when I was made in secret and skillfully wrought in the depths of the earth, probably a metaphor for the womb, the uterus. Your eyes have seen my unformed substance, and in your book were all written the days that were ordained for me when as yet there was not one of them. The psalmist doesn't know anything about pre-implantation zygotes, about pluripotency, about gastrulation, none of that. But he does understand something important. Inward parts, well, that's the Hebrew word rakia, or kelia, kidney. So my internal organs were formed before I was born. He understands that much of the process. You wove me in my mother's room. I was skillfully wrought. Rakam refers to the weaving of tapestries. This is a process of weaving that he understands. And your eyes have seen my unformed substance. That's the word golem. J.R.R. Tolkien fans, probably he got his name for Gollum from this word of unformed substance. So the psalmist understands that development is a process, and yet God was present throughout it. There was no place where the psalmist has ever been, including in his mother's womb, where God was not. Perhaps even more mind-blowing for Christians is the profound understanding that Christians have, at least historical Christians, about the second person of the Trinity, God the Son. We say in the Apostles' Creed, Jesus was conceived by the Holy Spirit and born of the Virgin Mary. He underwent embryonic development just as you and I have. Lucy Shaw, musing about this, said it this way in her poem, Made Flesh. After the bright beam of Annunciation fused heaven with dark earth, his searing, sharply focused light went out for a while, eclipsed in amniotic gloom. His cool immensity of splendor, his universal grace, small folded in a warm, dim female space. You see, the second person of the Trinity has graced our story and indeed the entire process of embryonic development by engaging in it. That should lead us to a sense of wonder. Well, we've come to the end of the story. Thanks for listening. I hope you've gotten a sense for the amazing self-construction project that embryos are, that each of us was as we developed. And I hope you've enjoyed learning a few things about what we understand as biologists about how those processes work and how we might think about them from the perspective of faith. Thanks very much, and I'm happy to entertain questions.
Well, thank you very much, Professor Hardin, for a very instructive and illuminating talk. Actually, I learned a lot today, stuff I didn't know before. So it really was wonderful. Thank you very much indeed. So could I encourage you to send your questions in online? And if there are any questions here in the audience, then as I said before, please come to the front. So we have one question that I could go to, first of all, from one of our online viewers, who asks if there's essentially a cutoff point in the embryo's development when it can be identified as a person. And how do you, as a Christian scientist or scientist or Christian, think about this issue about when the embryo actually can be considered a person?
Yeah, that's obviously a key question and a great one. I'm a biologist, so I will speak first from the perspective of biology. I think the biological sequence of events, as I've laid out some of it in thumbnail form today, is beyond doubt about how each of us normally develops. So that we can trace our beginnings from the one-cell zygote after the genetic material is mixed during a process known as syngamy in the one-cell zygotes. That's how each of us got our start. And so you and I as organisms can trace our life history back to that moment. So I think that's not in dispute. The question, though, is whether as the question phrases it, when do we have a person? That's a philosophical or theological concept. And the fact is, Christians disagree about this. Some Christians would say, well, it's not, that there's something added that is a non-material essence that is the sine qua non of personhood. Some people would view things that way. That gets challenging as we think about what the Bible seems to talk about, which is that we are kind of biological entities that are unified, both spiritually and biologically. But there are ways to think about that in various ways. Some people would say that important essence is added at fertilization. St. Thomas Aquinas thought, well, no, there's a series of Aristotelian souls that are added until you get to a particular kind of soul that's the one that underlies personhood, but that happens later. Other people would say, well, you need to have certain kinds of characteristics. And higher brain function would be one of the most important there, potentially. But there is a problem, and that is that each of us, when we were a one-celled zygote, has the potentiality to develop higher brain function if given the appropriate environment. So Christians simply don't agree about that point, whether we should take some sort of whether there are aspects of our biological functions that need to be present for us to assign personhood in this way. I tend to like a guy named Gilbert Mailander, and he would say, yes, Christians disagree about this, but if we're not sure, then we should err on the side of extreme reluctance. And so that would, I think that's a wise course of action. I think this is particularly important because now that we can make pluripotent cells without destroying human embryos to harvest inner cell mass cells using induced pluripotency, wow, there are so many things that are not ethically problematic that were before for people who hold conservative views of embryos. So I'm excited to see where the induced pluripotency technologies go in the future. Okay, Dr. Alexander asks a real softball question. So he says, what is your view about current experiments to create human-monkey chimeras? Thanks, Dennis. These experiments are underway. So Carlos Ispasua Belmonte and his group in particular has been moving some of these experiments forward. It's actually started off using a different mammal, pigs. And that's because people were seeking to make chimeric embryos, pig embryos, into which human cells have been introduced. And the pig embryos are compromised, so they cannot make a certain organ system. So only the human cells, which retain the capacity to make that organ system, would populate that organ. And so you might make chimeric pigs with human organ rudiments in them that could be used for xenotransplantation, organ transplants. But now I think Ispasua Belmonte and colleagues just seem to be doing this because they want to know what the limits of chimeric embryos are. There's grave concern about making chimeras that involve human brain tissue in primarily monkey sort of context. And I think you can understand why there would be a lot of concerns about that. I'm not sure entirely what the value of that particular set of experiments is in terms of human medicine, but it'll work. It's pretty clear from other chimeric embryo experiments that you might not get a normal embryo out of it, but those embryos will go quite a long ways into fetal development. So this is going to raise a lot of ethical issues for us.
Okay, thank you. I have a question here from our live audience, a real person here. Not that Dennis Alexander wasn't a real person, but we'd
like to take that up with him later.
Thanks, Jeff, for your talk. I have a background in science teaching, And in schools, the ethics of human embryos are not taught very well, I think for a number of reasons, partly that teachers are scared to offend different religious groups. I wonder if you can tell me, has this been your experience? And if so, what can we do about it?
So that is a really great insight. You guys have an additional complication in that there's religious instruction in the schools in the UK that we don't have in the US. I actually think that could be an advantage, and here's why. In my introduction to animal development course at University of Wisconsin, I devote about 4 lectures worth out of a semester to bioethics, because most of the students have not been exposed to it either in the US educational system. And I think as citizens of the world, they need to think well about these issues. The 21st century is a century of biology. And if our students are ill-equipped to think about bioethical issues, that is going to come back to haunt us when they are the leaders of our society. So I tell my students that we're going to do this, we're going to talk about ethics. And I point out that our ethical frameworks are derived from our worldview, our Weltanschauung, how we see the world, and that includes how we think about what's real. So ontology, how do we know what's real? Epistemology, metaethics, what's the nature of the good, and what is our ethical responsibility towards other people? So I lay out that those are fundamental issues, and then I simply state we don't all have the same worldview, and that seems to take a lot of the charged particles out of the room, allow us to talk about these things in a way that allows the students to be authentic. Some of the students, a minority of the students in my class, might be Christians, some might be Protestants, Roman Catholics of various sorts, others might be other kinds of monotheists, many would not have any particular religious persuasion. But I found that starting there is very helpful. And then to lay out, okay, once you think about various worldviews, what are the entailments of those worldviews in terms of thinking about ethics? And then deal with several case studies across a spectrum of views. That tends to set the stage in a way that allows all of us to value one another in terms of our worldview commitments, and gives us permission to talk about them. And I lay out that, in fact, I'm a Christian. And that, I think, helps some of the students to say where they're at. And then we can move forward and talk about them. And it also takes this away from the province purely of philosophy classes, which is where a lot of bioethics is discussed.
Okay, Professor Hardin, thank you very much indeed. I'm afraid we're sort of running short on time here. I was itching to ask you a question about how far we are from actually genetically engineering people, because I've always fancied having eyes in the back of my head, and whether that can be arranged in the future. But we're going to have to leave the questions there for the time being. Thank you very much again, Professor Hardin, for a wonderfully illuminating talk, and obviously stimulate a lot of profound thinking about the spiritual significance of some of these developments. And it's probably a good point at which to advertise this wonderful book, Wonders of the Living World, by our own Ruth Bankovich at the Faraday Institute. And you can buy copies online by going to the Faraday Institute website and clicking on the drop-down menu for the shop, Wonders of the Living World, which picks up much of the theme of today's talk. So thank you again for everyone who sent in questions and asked them in comments, and especially to Professor Hardin again for his talk and for the way he responded to those questions. And I'd like then just to finish by reminding you of our next seminar in 2 weeks' time, on the Tuesday, the 23rd of November at 1 p.m, when we'll have Professor Stephen Williams speaking on Thinking Machines, a Christian Perspective. So thank you once again, and we look forward to seeing you here in 2 weeks' time. Goodbye.